Hi everybody, Dr. Ahles here. In this short video, we're going to explore what happens to extra neurotransmitters after a message is done, and we're also going to look at the different kinds of synapses that neurons use to communicate with each other. Let's start with talking about extra neurotransmitters. When a neuron sends a message to its neighbor, it's only trying to send that message once. When I'm done hearing that message should go away. Otherwise, I'm going to hear that same message over and over again. So as we talk about these mechanisms here for dealing with extra neurotransmitters, the goal is to make sure that I hear the message once and don't keep responding to it. The first way that I can deal with neurotransmitters when I'm done with them is using an enzyme to break them down. There's an enzyme called acetylcholinesterase that breaks down the neurotransmitter acetylcholine. The way that this works is it chops it into two small pieces that would no longer activate the receptor for the neurotransmitter. This leads the message to no longer be heard. The second way that we can get rid of extra neurotransmitters is we can do a process called reuptake. Reuptake occurs when the neuron that spit out that message, the presynaptic neuron, actually grabs that message again. This saves the neuron some energy so it doesn't have to remake the message, and it also makes sure that it's not heard again so that the neuron only hears it once. We also have some neurotransmitters that just simply diffuse away. As they're moving away from the synaptic cleft, some of them will be taken up by the helper cells, the glial cells, and some of them will just get very diluted in the environment. So we want neurotransmitters to stay around only as long as they're supposed to. We hear that message and then we'll either chop it up, we'll take it back inside, or it'll diffuse away out of hearing range of my postsynaptic neuron. There are medications that change the way that neurotransmitters behave between two neurons. So there's a type of medication called an SSRI, and this is a medication type used to treat depression. SSRIs are selective serotonin reuptake inhibitors. Its name tells me what it does. It selectively inhibits the reuptake of serotonin. So when we talk about serotonin, this is one of the neurotransmitters that regulates things like mood or happiness. The job of an SSRI is to prevent serotonin messages from getting back inside the presynaptic neuron. This means that serotonin stays in the synaptic cleft for a longer period of time, and the longer that serotonin stays in the cleft, the more the body is able to detect it. So the mechanism behind an SSRI antidepressant is to help your body hear these serotonin happiness messages a little bit better. It keeps the message around in the cleft for a longer period of time. So one example of a way that I can modify the process of reuptake in a neuron. Everything we've been talking about with neurotransmitters is all happening at the synapse. So a synapse is just a fancy word, again, for a place where two neurons meet. Remember from our sketch that we have a presynaptic neuron. The presynaptic neuron is the neuron that brings the message to the synapse. The postsynaptic neuron is the neuron that's going to receive the message at the synapse. Remember that different parts of these neurons are found at the synapse. So the presynaptic neuron's axon terminals are at the synapse, whereas the postsynaptic neuron's dendrites are found at the synapse. Remember, this relates to the fact that one brings the message and one receives the message. The synapses we've been talking about up to this point are chemical synapses. And when we talk about chemical synapses, I want you to underline, highlight, star that they are an indirect method of communication. And what I mean by that is I release a neurotransmitter from one neuron and I send it across this space to the other neuron. It goes through the synaptic cleft to get to the neuron that it's talking to. We can give both excitatory and inhibitory effects at a chemical synapse. And I want to remind you from our previous video that some of our receptors for neurotransmitters are ionotropic. Remember that this means my neurotransmitter directly attaches to my ion channel, allowing me to change my charge right away. Some of my receptors, though, are metabotropic, and remember that a metabotropic receptor takes a little bit longer to have an effect. Both of these kinds of receptors are seen at chemical synapses. And at a chemical synapse, again, I release a neurotransmitter chemical, and it has an effect on my post-synaptic neuron. Excuse me. The other kind of synapse that our neurons use to communicate with each other are electrical synapses. And when we talk about an electrical synapse, these are formed with something called a gap junction. And it's been a while since we talked about cell junctions, but I want to remind you that a gap junction is literally a connection or a channel from one neuron to another neuron. So ions that I find in my presynaptic neuron literally travel through the tunnel into my postsynaptic neuron. Because these neurons are connected to each other, this is going to be a more direct form of communication. Electrical synapses are not very common, but we see them in places like the hippocampus, helping our neurons that are responsible for your memory. We also see them in locations in the cerebrum, which is the brain, involved in processes like eye movement. So different kinds of synapses used for slightly different functions. The last thing I want to mention about synapses is that not all of the synapses happen on the exact same place of the postsynaptic neuron. We've talked a lot about what would be called axodendritic synapses. Axodendritic synapses happen when the axon terminal in green of one neuron synapses with the dendrite of another. Axo for axon terminal, dendritic for dendrite. This is a very common kind of synapse that we see between neurons. We also will see several axosomatic synapses. An axosomatic synapse still includes that axon terminal where the message comes from, but this time that message actually goes to the cell body. Or remember, the other name for the cell body was the soma. So axosomatic synapses, like we see here with the red neuron, the axon terminal comes in and synapses directly with the soma, or the cell body of the postsynaptic neuron. The one other kind of synapse that we do see occasionally is what's called axoaxonal. And an axoaxonal synapse occurs when the axon terminal actually synapses with the axon hillock of my other neuron, or the very beginning of that axon. So this is putting information right into the source where we're adding messages together, where we're doing summation. With each of these types of synapses, make sure you know where they're found on the postsynaptic neuron. Use those hints in their name, whether it's part of the dendrite, whether it's the soma, the cell body, or whether it's that axon or axon hillock. Their name helps us to figure out where they're found.